Hello everyone, and in this video we'll be looking at protein digestion for a college athlete who's trying to increase muscle mass. Right off the bat, when protein is first ingested, it will start the digestion in the mouth through mastication, and in the chewing motion will break apart the proteins. Once the proteins have been masticated, they will make their way to the stomach, where parietal cells will release HCl or hydrochloric acid to increase the stomach acidity. After that, chief cells will release pepsinogen, which is an inactive form of the endopeptidase pepsin. And the HCl activates the pepsin, which is then used to break apart the polypeptide chains into polypeptides. Once they are brought into polypeptides, we move to the duodenum, where the polypeptides are then moved. S cells are then activated because of the hydrochloric acid, and then releases HCO3, which is the bicarb system, allowing I cells to activate to bring in protease. Protease is released from the pancreas, which brings pancreatic juices and other endopeptidases, such as chymotrypsinogen and trypsinogen. These things are also activated to chymotrypsin and trypsin. And in this, they are broken down into tripeptides and dipeptides. Finally, we move to the jejunum into a enterocell, where at the apical layer, aminopeptidases are used to break apart tripeptides into dipeptides, and dipeptidase is used to break apart dipeptides into amino acids. The first cotransporter we have is PepT1, where tripeptides and dipeptides are brought into the cell with a hydrogen molecule. Next, we have the Na amino acid pump, which is also secondary transport or secondary active, and this allows for amino acids to enter into the cell with a sodium molecule. Lastly, we have the NHE3 exchanger, which brings in a sodium and releases a hydrogen to keep the molecule gradient. And inside the cell, we have a intracellular protease, or peptidase, which is used to break down the tri and dipeptides inside the cell so that it can enter through a facilitated diffusion and into the capillaries. Once in the capillaries, there are a couple of ways it can move. One is through our amino acids, or essentially amino acids, where it can go to the muscle and begin muscular reconstruction and such. Also, the 10 essential amino acids are only brought in through meats and high protein vegetables and such. The 10 essential amino acids are phenylalanine, valine, tryptophan, threonine, isoleucine, methionine, histidine, arginine, lysine, and leucine. The other way amino acids can go is into either transamination or oxygen deamination. What we would like is to have 85 to 90% in transamination, where amino acids and keto acids are able to reproduce and make more amino acids in order to continue with muscular reconstruction. This occurs in the cytoplasm of a cell, in all cells, and in order for this to happen, we need a vitamin B6 to transfer O2 to a amino acid and NH2 to a keto acid in order to make amino acids 2s and keto acid 2s in order to enter with a nitrogen pool to make more amino acids. Now, for a collegiate athlete who is currently active, we'll then use the amino acid 2s to make glutamate in order to make ATP. When making ATP, the rest of the keto acid, or the glutamate, must enter through the cell and head to the liver, where glutamate dehydrogenase will break apart the NH3 and the keto acid into the TCA cycle, where it can produce more glucose in order to help with energy. The other direction is where the NH3 then goes into mitochondria to produce NH4 and then enter the urea cycle and finally enter into the capillary or into the blood vessel to go as urea through to the urethra and then be excreted as a urine. Thank you very much for watching, and have a great day.